And I'm going to talk today about using individual-based simulations to study adaptation and genomics. So like a lot of the talks today, kind of interested in genomics and what we can do with this. So we can go to the first slide, Jenna. Okay, so broadly, we can find evidence of local adaptation just about anywhere we look in nature. And people going out in nature, looking at plants and animals, at sticklebacks, as Sean was talking about before, at mice, have found that species are often kind of adapted to their local environments when there's different environments from one place to the next. And so there's divergence between populations. And on the next slide, we can see there's been a lot of interest recently in looking at the genome for evidence of local adaptation, and to see what kind of signatures might be characteristic of local adaptation. And as I've shown you here, there's a few different kinds of approaches people are using to this, but generally what they're trying to find is signals of extreme divergence between different populations that are characteristic of the kind of thing you'd expect if there's local adaptation happening. And there's a bunch of different statistical ways we can go about looking at this in the genomic level. And there's just been this broad kind of push to try and get genomic data into the kind of study of adaptation. And as Sean mentioned, we're now kind of at this point where we really need to kind of expand the way we use this data, both through experimental approaches and through our kind of statistical and theoretical appraisals of what's actually happening. So we can go to the next slide there. So to do this, to introduce this problem, I thought I'd use the stickleback, which is a nice species and a nice example, and one that Sean already nicely introduced this morning. As we talked about before, the stickleback have this kind of distribution all around the world where they've diversified over and over again as they've colonized fresh water, and there's these kind of freshwater ecotypes and marine ones that differ in plating and morphology and all sorts of other physiological and morphological traits. And the study by Felicity Jones that Sean talked about this morning was looking at divergence between repeated pairs of populations that are inhabiting freshwater versus saltwater. And if we can go to the next slide. And Sean showed this slide as well, a nice overlap here. This is a really nice example from Felicity Jones's paper showing these kind of genome scan, looking at the loci that were highly divergent between these freshwater and marine populations. And as Sean pointed out, there's lots of these kind of green dots or these peaks that are implicated in divergence between populations. And it's really interesting when we see that divergence tends to line up with genes like EDA, which we know is involved already from other work, involved in the actual phenotype of plates. But then, next slide, we can see there's actually a whole lot of other loci involved in the genome that we don't really have much of a physiological or a mechanistic reason to understand what's actually happening with these loci, but we want to know what's going on with them. And part of the problem with interpreting genomic data is that it's not always clear whether or not these kind of statistical outliers are outliers due to selection, or whether there's just some other kind of funny stuff going on in the genome. And when we're testing so many different loci in the genome, there's a lot of chance for funny stuff to go on. And so, next slide. What I'm really interested in doing is kind of dealing with this challenge of how we interpret patterns in this big genomic data, given that we have some background. We've got some nice long history of population genetics and statistical tools we can do this, but we're now starting to look at more complex traits and realize that there's some bigger problems here. So as I'm kind of showing here, some of the interpretation of genomic data is quite simple when we have loci that are kind of more or less like simple genetic basis traits, where there's just a very large locus underlying a trait and a strong selection on that trait. It should conform pretty well to classical population genetics, and it should have well-behaved properties that fit well with nice statistical tests. But actually studying patterns is much more difficult when we have more complex or maybe more realistic cases for a lot of biology, where there's traits that have a very complex multi-locus basis, and there's been more changing demography. We don't have this kind of nice stable equilibrium population. And so the reason I kind of say all this is that individual-based simulations over the past few years have been used in a lot of work that's helped highlight some of the difficulties involved in these more complex cases that don't reduce quite so nicely to the single locus predictions that we get. So I'll move now into kind of talking about a few examples of how individual-based simulations have shown some interesting patterns, and then talk specifically about how else we might use them. So next slide. So one of the first problems in trying to deal with any kind of signature of local adaptation is trying to distinguish alleles that are under selection from alleles that are just neutral in the background, in the kind of overall genome. And the problem with kind of complex traits, multi-locus traits, is that as the number of loci increases underlying these traits, the strength of natural selection acting on each locus is necessarily going to decrease. If you have the same strength of selection in the phenotype, but it's spread among more loci, there's less effective selection on each one. And some nice work by LaCourrier and Kramer using individual-based simulation showed that as you have the adaptive loci in the dark gray in the middle, and you compare those to the neutral loci in the rest of the genome in this kind of medium gray, you can see that as you go to a larger and larger number of loci underlying your quantitative trait, there's a smaller and smaller difference between the neutral loci and the quantitative trait loci. And so even though there's lots of divergence at the phenotypic level, which is that light bar, it can be very difficult to distinguish selected loci from neutral loci as we move into this realm of kind of highly polygenic traits. And so individual-based simulations were used in this case to show this nice qualitative problem that we're going to be facing as we deal with genomic data. Next slide. So some work that I've been working on recently was looking at this kind of issue as well, but looking at what happens where instead of changing the number of loci underlying a trait, we're changing the size of the alleles that are involved. And so these plots here are showing what happens when we just have large effect alleles that are contributing most of the divergence to a single phenotype. On the left-hand side, you can see there's this phenotypic divergence is plotted over generations. And a lot of divergence between trait means is just showing that there's two different populations that have adaptively diverged, and there's a large amount of phenotypic difference between them. And on the right-hand side, the blue lines there shows the loci that are contributing to this divergence, and you can see there's this relatively stable genetic architecture where there's 5 or 6 genes or loci that are contributing most of the divergence between populations. And this is kind of what we imagine we're going to find when we kind of have the best case scenario of looking at the genome. This conforms to the classic expectation where we're kind of thinking there's a nice stable architecture underlying this local adaptation. But this isn't always the case. And so in the next slide, you can see in this one here, now changed it. So now we just have lots of small alleles of small effects. And so here, what we're looking at is now there's phenotypic divergence still on the left-hand side, but what you can see on the right-hand side is there's just this kind of wash of divergence contributed by these different loci underneath. There's no single locus that's contributing divergence for very long, and there's just this transient genetic architecture. So even though there's a stable-looking phenotype, what that phenotype is made up of changes almost from one generation to the next. It's changing on the scale of a few hundred generations. So we're seeing very rapid change in the architecture of divergence despite apparent phenotypic stability. And so this highlights another real problem if you're trying to look at the architecture of the genetic divergence is there's not really anything that's stable in terms of genetic architecture. So just another kind of issue that's individual-based simulations can help us show that isn't immediately obvious from analytical theory. Next slide, please. So a final problem that's been really nicely highlighted through evolutionary individual-based simulations is looking at the issue of non-equilibrium demographics. And so here I've just showed a particular species that was expanding from glacial refugia after after the glaciers retreated, and these expansions, next slide, can be associated with a phenomenon called allele surfing. And this is where as a population is spreading to a new environment, it's growing and kind of colonizing through a process of repeated bottlenecks. And because these repeated bottlenecks can greatly enhance the effective drift, you can get very large changes in allele frequency just due to drift. And on the next slide, you can see some simulations done by Escoffier and Ray showing what happens in a typical kind of surfing scenario, where you've got population expansion from the middle of each of these plots. And then what you can see is that the black or the white shading reflects which allele is actually present at high frequency. And so you can see here, if you were just going to sample alleles from the margin of this species range, you would see very strong differences between which allele was present only as a result of where this surfing phenomenon had happened. And so individual-based simulations here showing, you can get these signatures purely by neutral alleles that look a lot like what we would kind of classically think is adaptive divergence, where there's strong differences in allele frequency. So next slide, please. So I think what these and many other simulations that have been done recently have showed is that we're really only scratching the surface of understanding how evolution can shape patterns of the genome that look like local adaptation. And so one thing that I'm broadly interested in is how can we use individual-based study simulations to kind of help interpret genomic data and kind of further this kind of quest to understand what's going on. So next slide. Individual-based simulations, what I mean broadly is just simulations that have rules about how an organism is built, what kind of genetic loci, the organization of these loci in the genome, and other life history details, and then also what environment they're colonizing, what the structure of space looks like, whether it's a populations or clines, and then rules for the life cycle that say how organisms should go about reproducing, surviving, and inhabiting this kind of virtual environment. And so what we do is we just allow digital organisms to reproduce and then study how they evolve over multiple generations. Next slide, please. So, I think there's a lot of different ways that individual-based simulations can help us understand genomic data. And they're a nice complement to analytical theory, where analytical theory is incredibly powerful because you've got a nice mathematical model where you can just plug in numbers and get an immediate answer, but it's more constrained. And so individual-based simulations are nice because they allow you to more flexibly explore the way evolution works. You can look at things like multi-locus traits with very complex architectures, non-equilibrium scenarios, or look at haplotype scale patterns and associations among loci much more easily than you can with an analytical model. Next slide, please. So I think I've kind of grouped how I think individual-based simulations can help us into kind of two broad groupings. One is kind of applying them in a specific context to test hypotheses about a particular pattern in a given species. And you can use this either as a kind of a positive proof to test whether an observed pattern that you see in nature is consistent with a given model, or as a negative proof to see, try and narrow the space that we understand things, whether a particular pattern is inconsistent with a given model. And I'll go more into this in a second. The second broad part is to ask, a general method, use simulations to broadly understand how does evolution work, and can we kind of formulate qualitative hypotheses about evolution that should kind of give us these kind of broad tests, or just help us understand more clearly what general patterns we expect to see when we look in nature. Next slide, please. So as an example of a kind of an empirical application for simulations, thinking about going back to these stickleback, we might try and simulate a scenario that looks a bit like the stickleback, where there's freshwater and marine populations, by simulating, say, two patches with migration and divergent selection, where maybe, say, red and blue are like the saltwater or the freshwater. And we might want to ask, how much migration or how strong does natural selection have to be to yield a statistically detectable response at, say, 50 loci? So trying to understand what's happening on the right-hand side in this genome scan using these kind of simulation models. And that's an interesting problem. But as you see in the next slide, this kind of application requires that we make all sorts of assumptions about things like allele frequencies, mutation rates, the sizes of beneficial mutations, population size, and demography. And so things like the amount of time it takes for adaptation to actually occur. And so when we're actually running simulations, we either need to guess at what these things should really be, or take measurements from what we think they are in nature, or we have to simulate across these very wide ranges of parameter space to try and do this. And So for every unknown parameter that we add into simulations, the kind of span of plausible results grows, and it gets very difficult to say anything conclusive. And so broadly, one thing I would suggest is that I think negative proof coming from simulations is much more compelling than positive proof. What I mean by this is by saying something like, if you run a few simulations with a bunch of migration rates or a bunch of strengths of selection and find that these particular regions of parameter space are inconsistent with your observations, then at least you've learned something a little bit more concrete. Just showing that one particular simulation is consistent with results isn't as compelling, I would say, because there's just so many other kinds of parameters that might yield the same kind of result. So there's a lot more I could say there, but I'll move on for now. So the next slide, please. So I thought I'd end up by talking a little bit about technical considerations. If you're going to get into running simulations, the kinds of things we need to consider and why it matters. And so kind of each one of these things that I've written on the left-hand side goes with all of the things on the right-hand side, needing to consider things like, what regions of parameter space do we need to consider to answer our question? How many replicates of simulation should we run? How long should we run them for? And what kind of initialization conditions should we do? And so I'll consider each of these kind of in the next few slides. So I'll move to the next one. So obviously, if we had infinite wisdom and power and computers, we'd just want to maximize parameter space. We'd want to just simulate everything and get as much data as we could. But of course, that's not going to work. We've got computational constraints, and we just got limited brain power. We can only analyze so much data before our head just explodes trying to look at it all. And so in the next slide, I've just shown some kind of broad computational costs to give an idea of how much these simulations take. The simulation program that I've used a lot in my own research called Nemo, its computational cost scales with the number of generations you run it for, the number of individuals you put in the simulation, and the size of the genome, the number of loci that you put into that genome. And so using kind of typical lengths of simulations that I've run with 2,000 individuals and 1,000 loci and 1 million generations, it takes about 200 hours to run one of these simulations, which is a long time. But when you really look at it, this is simulating 5 million loci per second. So it's screaming fast calculations running this thing, but when you're trying to simulate deep time evolution, it's just a lot of calculations to run. There's also RAM constraints. So basically, if you're going to simulate a population, the computer needs to hold all of that in memory. And so in Nemo, a typical kind of cluster run with about 2 gigs of RAM will max out at about maybe 1,000 loci in about 10,000 individuals. So that's about the maximum, number of alleles per population that you can run, depending. And you can simulate more individuals with fewer alleles or more alleles with fewer individuals. But that's kind of gets towards an upper bound when you run these things on a computer cluster. Next slide. And this of course butts up against the problem of how broad do you want to simulate if we're for example simulating adaptation to climate change, and we've got simulations that take about 2 days to run. If we start off by just say exploring variations in the strength of selection, with 5 different settings for the strength of selection, this is going to take 10 computer days, maybe split apart on a cluster, but quite a while. And then if we add on different each new parameter that we add on, if we're going to study all possible combinations of those parameters, multiplies the number of days we're going to take. So here, just a simple kind of cartoon example, if we add on 4 parameters with a few different combinations for each one, we'll end up going from 10 computer days to 1,000 computer days and rapidly get towards something that's kind of computationally very slow or intractable even. So computational costs become a real thing to consider when you're trying to approach the problem of how you use individual-based simulations, which is very unfamiliar if you're used to working with things like coalescent theory or with mathematical analytical models, where you can just plug numbers into an equation and get a nice answer out. So next slide, please. So of course, analysis and visualization is a big problem. And if you simulate something like, say, 10 different variables, how do you study the results? It gets incredibly difficult to graph a 10-dimensional space. I certainly can't visualize it. I kind of max out at about 1 or 2 myself. And these are one way that I've addressed this kind of thing before is just printing out lots of graphs and sticking them on a wall and staring at them. But it's visualization problems on the next slide are a real issue that we're kind of going to have to confront in the coming years. And I'm hoping that as more people use simulations and kind of get into doing this for studying evolution, we'll find out some more interesting ways to kind of try to visualize these highly multidimensional data sets. Next slide. I'm kind of rushing through these things because I think a lot of these are practical concerns that are fairly obvious. So I'm happy to talk more about any of them later on. What I've shown here is just a cartoon example of some data. This is real data that I got from some simulations I ran a long time ago. But it's just looking at, say, some evolutionary factor of interest, how much variance is maintained in a simulation as a function of migration rate. And we can see, at higher migration rates, there's a lot more genetic variance maintained. And this is kind of just illustrating what happens when you've kind of the importance of exploring parameter space. So on the next slide, I've drawn that curve in here in green on this slide. And what you can see is that, that where we might have concluded, oh, there's a very big effect of migration rate on this genetic variance, that's actually highly dependent on other parameters. And so in this case, mutation rate is plotted along the other axis. And as you'll see in the next slide, there's really actually quite a small effect of migration on the maintenance of genetic variation when we've got, small mutation rates. And so the biology isn't so important here, but what I'm trying to show is just that even for two variables, it's really important to kind of bracket all the parameter space you can so that you can see whether there's an interaction between them. But when you go from two variables into 5 or 6, visualizing them and trying to see whether or not there's kind of complex and interesting interactions becomes incredibly challenging, and but very interesting. I mean, that's exactly why we're trying to use these things. So, next slide, please. A final kind of technical thing that's important is the span and resolution of parameter space. So again, this is real data just on the biology doesn't matter, but it's looking at allele effect size as a function of migration rate in some simulations. And here I cherry-picked a few parameters. If I'd run a sparse data set here, we'd kind of expect some kind of monotonic reduction in allele effect size as we get increasing migration rates. But on the next slide, you can see if I'd actually run a higher density coverage of parameter space, there's a completely different and very non-monotonic pattern that pops out. And so just another kind of issue that comes in when you're dealing with costly simulation runs, each one of these takes a long time to run. You want to kind of make sure that you get enough coverage of the actual parameter space that you're going to see interesting or realistic patterns at a fine enough scale along parameter space. And it's easy to sometimes miss these kinds of things. Next slide, please. I think I'll skip this one. This is just saying it's really important to run lots of replicates. So next slide after that. Another factor that I've run into if you're ever conducting simulations is just how long to actually run a simulation. And this slide shows what looks like an apparent equilibrium that's been reached at the amount of divergence between two populations run over a long period of time. These are 50,000 generations and looks like it's kind of leveled off to some equilibrium. But as you can see in the next slide, this is exactly the same run of the simulation just carried out for another 450,000 generations. And you can see now it seemed to have reached a different equilibrium, a slightly higher amount of divergence after a long period of time. And if we go to the next slide, you can see, in fact, there's just been this very, very gradual trend in increasing divergence over a very long period of time, and that even the true equilibrium of this simulation hasn't been reached after as many as 500,000 generations. And so I show this just to say that it's really important to be aware of the rates at which important processes, evolutionary processes, are running. Because in this case, what's going on is there's a long-term evolution of the genetic architecture underlying this phenotypic divergence. And so the phenotypic divergence evolves very quickly to something close to the equilibrium, but then this secondary evolutionary process that happens with genetic architecture is occurring and results in this very gradual change in divergence. And so running simulations, it's very important to be aware of the kinds of things that might be happening underneath and really consider whether or not something that looks like an equilibrium really is an equilibrium. Next slide. I think I'll skip this too. This is just saying that initialization conditions are important. So next slide. I think it's just really important to be aware of the kind of ways that the biology matters and the initialization parameters that you might set in your simulation affect the way that evolution works. So we can skip to the next one. Just that I think with initialization conditions, what kind of settings you run for a simulation really affect the answers you're going to get from it, and trying to use analytical results or some kind of classical population genetics to help understand whether or not you're going to have, kind of complex equilibria, whether or not the equilibrium that you find is going to be a product of the initialization condition that you set is very important. And one approach that I've seen sometimes people do with running simulations is just to actually randomly generate thousands of different parameter combinations across, the whole region of parameter space to just try and get a broad look at, if everything is equally possible, then what is evolution going to do? And that's one interesting way to run things for empirical applications, but it's not particularly satisfying if you're trying to really understand why evolution works the way it does. Next slide, please. So I've kind of alluded to this before. I think there's two really important things I'd mentioned biologically that I've found over doing lots of individual-based simulations, is that running variables that have very low rates of occurrence, things that happen less than 10 to the minus 5, that should say, not 10 to the 5. So things that are very, very slow, rates of mutation and rates of recombination, for example, can take a very long time for the simulations to equilibrate if these kind of factors are under weak or indirect selection. And finally, because population size affects the rate of mutations that are occurring in a population and the availability of beneficial mutations in a population, the overall rate of adaptation also scales with population size. And so it becomes very difficult to study things like rates of adaptation when we're constrained to running small population sizes because of computational constraints. So things to be aware of. Next slide, please. And so I think this kind of dovetails into some of the challenges in future directions where I think I'm really excited to see what people that are more savvy with computers than I am come up with, ways to increase computational efficiency to allow us to look at things with larger population sizes and explore the effect of drift on evolution. Simulating more realistic landscapes, things that are more spatially explicit or have more realistic kind of geometry of the landscape and demographies. And as I was mentioning before, this visualizing high dimensionality results is a real problem. And I think by making animations and other things where we can plot graphs over time helps us kind of stack things in other dimensions. It's a nice way to think about moving forwards with that. And finally, I think there's been some really neat work done by people like Jeremy Draghi looking at biologically realistic models of things like gene interaction networks, and looking at how actually simulating explicitly these kind of details in a simulation can actually really quite change the way that adaptation works. And so with that, I think I'm finished. Last slide. I'd like to acknowledge all sorts of people that have worked with me on simulations over the years I've been doing this, and I'd like to thank you for listening, and Jenna for inviting me.
Hi, Sam. Good stuff. I had a question for you that I've been chatting with a few folks here in Calgary about at the meeting today. One thing about your individual-based simulation approach is that it gets away from the classical population genetics approach of the way we set parameters, like population size is a parameter, selection coefficient is another separate parameter, right? Whereas in your individual-based simulations, things like populations, at least some of those parameters end up being, the traditional parameters end up being interconnected, because actually in your individual-based simulation, you're making some kind of assumption about the underlying biology that then, determines as an outcome both the population size and, other parameters or something like that. Do you see what I mean? And
yep,
so I'm wondering if
sure.
you could just comment on how that, presumably that both creates the potential for us to discover new phenomena we wouldn't have discovered with models, at least not very easily, with models parameterized in the traditional way. And maybe it also gives either opportunities or creates obstacles for simulations, maybe in terms of, like how many parameters you need to simulate
Definitely.
a given system?
Definitely. Yeah, so the work I've done with Nemo Nemo is basically built to try to mimic the way population genetic models work as closely as possible. And so it kind of does population regulation in the same way that soft selection models do regulation. And it tries to do selection in the same way that, say, a viability selection model would do it. So it tries to emulate population genetic predictions as closely as possible. And so it tends to be quite close to PopGen models, and it actually behaves pretty much identical to them. So we can plug in the results from Nemo, and they basically they mimic exactly what we expect often when we do these kind of, compare them to approximations like, say, Kimura's fixation probability, or, the Reinhard-Berger has an approximation for how much genetic variance should be maintained at equilibrium, and that fits quite nicely with what we see.
So then again, the question could be reframed as, is it interesting to try to get away from that?
Yeah, so that was what I was going to get to then, is that, I think that basically we started off like that because we wanted to kind of use the analytical theory as a guidepost. But really, the analytical theory and the assumptions of PopGen are quite constraining, as you mentioned. And so I think that, having really variable fecundity, or having really variable, having population sizes that are determined by the intrinsic fitness of all the individuals within that, or those kinds of more realistic, more, say, ecologically motivated choices, I think could be really interesting. And I think I've seen some really interesting work where, simulations were made spatially explicit, where every individual had a particular spot in space, and that actually totally changes the way, a speciation model works. So I think there's so much to be done in terms of relaxing the assumptions of PopGen models and seeing what happens.
Okay, thanks, Sam. We're just keeping an eye out here to see if anyone else has any other questions.
All right, I think now. Thanks so much, Sam. I'm going to hang up on you now.
Okay, thanks, everyone.
It was great talking to you. Thanks for tuning in.
See you soon.
Bye.